So, good evening, dear participants. Welcome again to this 1 month plant genome workshop jointly organized by Decode Life and Ensemble team from EMBL, European Bioinformatics Institute. We are honored to have the esteemed Ensemble team from EMBL, European Bioinformatics Institute, UK, to join us for this event. Your expertise in bioinformatics is invaluable to our participants, and we expected that our participants will learn from your experiences. So, without further ado, I hand over the online session to Ensemble team. Thank you for being with us today. Over to you, Lucy, for today's session.
Thank you very much. Hi, everyone. Welcome to the Ensemble Plans browser workshop. I hope you've been enjoying the course so far. We've worked with Decode Life in the past, and we always enjoy delivering these courses with Decode Life. So first of all, there are some construction works going on. So if my background noise is too noisy, please let me know so I can change the settings of my microphone. I hope you can see my screen. If not, please let me know as well. But I think we can begin in that case. So my name is Louis, and I will be delivering most of the workshop for Ensemble. I will also be joined by my colleague Ian Sang, and he will join us on the last day of the workshop where he'll talk about Biomart, which is one of our most popular tools in Ensemble. So the entire workshop is going to follow the following structure. So I will begin each module that will cover with a presentation explaining what the data is or what the tool is and how the data is being produced and what you can use the tool for. Next, I will give you a live demonstration on my screen. So this is where I will walk you through how you can find the data on the Ensemble browser. All you will need for the workshop is an internet browser. So anything like Firefox or Chrome will work perfectly with Ensemble. And then after the live demonstration, I will give you some time to do some exercises. So this is where you'll get a chance to explore the data yourself and browse the Ensemble database and see what kind of information you can retrieve. So all training materials are available on the training website, which I will show you now. So I'm going to switch my my window, and I'm now on my internet browser. And you want to go to training.ensemble.org. And that's ensemble without the E at the end. I will also post a link in the Zoom chat in a minute, but I just need to show you where to find it first. So this is the homepage to our training website. At the very top in the navigation bar, you can find a link that says training materials. So I want to click that link, and our materials are available in this very first link that says Decode Live. And let me share the URL with you. All right, so this is the training website or training page that we're going to use throughout the workshop. On the right-hand side, we've got my presentation slides. So this is my PowerPoint presentation that you can download and review in your own time. Then we have a link to what we call the living document. I'll open this in a new tab just to show you how this looks. So this living document is very useful, especially for virtual training, because here you can find more information about the workshop. We have the agenda in the second page, some links to some important pages. But if you scroll down to page number 4, we've got a section for questions. So if you have any questions at all, you can ask them on this living document. You can enter your question up here, and then I will answer just below if you have any. So this document is very useful because it will be available to you indefinitely. So that means that if you had a question and you wanted to come back to the answer in 3 months, this document will still be available for you to refer to. So it's a nice kind of reference document for the future as well. If you prefer to ask your question on Zoom, that's fine as well. So just please raise your hand, and then I will ask you to unmute yourself. But I think in the case of our course, uh, if you ask the questions in the living document, I think this will be very useful, especially for future reference. So I do highly recommend using the living document for questions. And I will put a link into the Zoom chat as well. All right. So I'm back on the training page. At the very bottom of the page, we have the section called Demos and Exercises. And this is divided into the different modules that we're going to cover. So we're going to look at species and genome assemblies, genomic regions, and so on. So after each module, after the demonstration, you'll have time to do exercises. So if you open, let's say, our first module, we have these two options available. One is demo. So these are just screenshots of what I'm going to show you during the demonstration. And underneath, we have exercises. So these are the exercises that you're going to work on when I tell you to. If you have any questions at all, please let me know. There are also some answers down here. So please remember, this is not a test. These exercises are for you to explore Ensemble and become familiar with the browser. And I suggest you leave this website open for the rest of the course or for the rest of our sessions, because we're going to come back to it quite a lot in the next couple days. All right. So I'm back in my presentation now. So what do I hope you will learn at the end of the Ensemble workshop? So I hope that you'll have a good idea of what types of genomic data you can retrieve from Ensemble plants. We're going to navigate the Ensemble website, and we're going to explore what type of species we have available. Hopefully, your species of interest is one of them. We're also going to look at the different data types that you can access. So for example, the variation data, the comparative genomics data, and so on. I'm also going to show you the different ways of accessing Ensemble data. So we are mainly going to explore the Ensemble browser, but you can also integrate Ensemble data into your pipelines via our MySQL servers and REST API, and I'll talk a bit more about that during the presentation. And most importantly, after the workshop, I hope that you'll be able to know where you can find help and where to find the documentation pages in case you had any questions about the data that we provide. So in the next 4 days, we have 1.5 hours each day, and we're going to cover quite a lot. So hopefully we'll be able to get through everything, but if not, that's fine too. This course is there for you to become familiar with Ensemble plants. So if you want me to slow down, please let me know, and I can slow down, of course, and go through things a bit slower. But I do hope that in these 4 days that we have, I will be able to show you the Ensemble website, how to visualize genomic regions in the browser, how to look for genes and transcripts. We're also going to look at the different comparative genomics data we have available. So these are gene trees, homologies, and so on. We're also going to cover what type of genetic variation data we have, and we're going to look at one of our tools, which is the Ensemble Variant Effect Predictor, or VEP. So this tool allows you to explore the consequences of your own variants. And on the final day, as I mentioned, we will be joined by Ian, my colleague, who will talk you through Ensemble Biomart, which is another tool but this one allows you to export large amounts of ensemble data without needing to know any programming knowledge. At the end, I'll wrap everything up, give you a summary of what we've learned, and I'll also ask you for some feedback for the course. And this is also where you can opt to retrieve a certificate of attendance if you wish to have one. Okay, so let's begin with an introduction to ensemble plants. So just a few words of how the ensemble project actually started. So in 1977, Fred Sanger and his team sequenced the very first genome. This was a bacteriophage. And this really kickstarted the era of sequencing and sequencing much more complex organisms. So the very first draft of the human genome, for example, was published in 2004. And this was a project that was started in the 1990s. And since then, many, many more organisms have been sequenced. Today, we can sequence an entire human genome in a matter of days. So we've come a very long way. And with the publication of this human genome, the Ensemble project was launched in 1999 with the goal to automatically annotate the first draft of the human genome, but to also integrate it with other data and make it publicly available. But why do we need genome browsers like Ensemble? So with sequencing becoming much cheaper and faster, as you might notice that genomic data is piling up very quickly. But when genomic data is produced, you just get something like this. So this is just a file with a bunch of letters of A, T, C's, and G's. So if you tried to read a genome without annotation data, it would be like trying to read a new language without a dictionary. So it wouldn't really mean anything to you. And this is exactly what genome browsers are there for. Genome browsers give value to a genomic sequence. So what they do is they map annotations to genomic data. And Ensemble in particular brings together information from a wide range of other databases into a single site. You might have heard of other genome browsers like UCSC and NCBI. Just know that these genome browsers differ in the annotation methods, so the algorithms that we use. I also want to emphasize that Ensemble does not do its own sequencing. So we simply take publicly available sequencing data and we use our own pipelines to annotate the genomic data. The Ensemble browser is dedicated to vertebrate species, but as we know, vertebrate species are only a fraction of the different organisms in the world. So you can find some non-vertebrate species in the Ensemble main site. So for example, fly, worm, and yeast genomes. These are mainly there because they're commonly used model organisms. But in general, if you want to access any non-vertebrate genomes, you want to go to our sister site, which is Ensemble Genomes. And this is divided into different divisions. So we've got a separate site for plants, one for invertebrate metazoa, bacteria, protists, and fungi. And we also have a separate portal for the coronavirus genome. You'll notice that they have different color schemes, but the layout of the website itself they're all very similar. So once you're familiar with one division, one website, you'll have no problem navigating any of the other Ensemble sites. So Ensemble Plants has genomes for over 100 different species. We have gene builds available. So gene build is a genome browser specific term. It just means gene annotation data. So we have gene builds available for all these species. We also have comparative genomic analyses available. These include alignments, gene trees, and homologs. You can explore the genetic variation data we have available for a number of different species and use the ensemble variant effect predictor, or VEP. So we're going to explore this on day 3 of our course, of our workshop. You can also use ensemble to display your own data. And we have a number of different tools that you can access to use for your analyses. So I mentioned that there are different ways to access ensemble data. So we are going to focus on how to access it via the browser. But if you want to integrate ensemble data into your own pipelines, for example, you also have programmatic access via our APIs and MySQL servers, and the ensemble code and the ensemble data is completely open source as well. So if you wanted to use the ensemble data for your analyses, you are free to do so. Just keep in mind to cite the most recent paper of ensemble. And I'll show you which paper this is towards the end of the workshop. So we know we know that genomic data is constantly evolving and changing. And this is why ensemble is updated about every 3 months to represent the most recent findings. And behind every update, a lot of work is involved. So we add any new genome assemblies, we update underlying software, we update any gene annotation if there are any updates to be done, the variation data, the comparative genomics analyses, and we add any new interfaces if we think that this is appropriate. So the current ensemble plants release is 59, and this was published back in May. And around August or September time, we're expecting the next release, which is Ensemble Plan 60. And this is why, or the reason why I want to highlight this is because it's very important to write down which release you are working with. So if you wanted to access an older release, for example, let's say you want to access Ensemble Plans 59 in 6 months' time, you can do that. We do keep our archives open for up to 5 years, and then we take the website down, but the data is still available through our FTP site. So to keep your research consistent, make sure that you write down which release you're working with and that you work with the same release throughout your workshop. We also have what we call our Ensemble Rapid Release website. So this is another Ensemble site, but the difference to the Ensemble main site, Ensemble and Ensemble Genome site, is that this website is updated about every 2 weeks, so much more frequent. And because we update this much more frequent, the data is also more limited. So the only data we have available over the rapid release website are genome assemblies and gene builds. And the only other tool that you can use is BLAST. Keep in mind that the genetic variation data on rapid release is very limited, but it is being constantly updated or added, and the comparative genomics analyses as well. If you don't find your species of interest in Ensemble, you might be able to find it over the Rapid Release website. And if you can't find your species of interest at all in Ensemble, you can always message us and or leave a comment and ask for the genome to be annotated by us. Okay, so I just want to do a quick recap of how genome assemblies are generated, because this is going to be really important to understand the genomic regions view in Ensemble. So on my slide, what you're seeing is a simple diagram of sequencing reads. So each of these lines, each of these kind of fragments are sequencing reads. And sequencing data is assembled by matching up any overlapping reads. So if you take the reads and put them together, they will match up kind of like a puzzle. So what this makes up is the contiguous sequence, or what we call the contig. And a contig is just a consensus region of DNA. So these contigs are then assembled into scaffolds. Those scaffolds are just a run of contigs or an assembly of contigs without any gaps. And then finally, if there is enough mapping information, scaffolds are assembled into chromosomes, uh, which is the most complete sequencing kind of status that a genome can have. So the reason why I'm mentioning this is because you are going to come across the word context quite a lot in Ensemble. And the main thing to remember here is that when you see context in Ensemble, this is just referring to the sequence itself. With that in mind, genome assemblies are still not a perfect representation of the genome. So just like older versions of a world map, there are still some gaps in it. Um, and it's important to remember that there are still regions in the genome that can have errors and gaps, but these are being revised constantly. All right, so what can you use Ensemble for? To summarize, you can explore genomic regions, you can find information about specific genomic features in the genome, you can learn more about genes and transcripts of interest, learn about genetic variants in different species. I'll show you how you can find out if there is variant information available for your species of interest in Ensemble. Ensemble also has a number of tools that you can use. So we've got the variant effect predictor that allows you to find out the consequences of your own variants. We've got Biomart, which allows you to download loads of Ensemble data in bulk. We also have Blast and Hammer for any sequence or protein searches that you might want to do. We have a few species available for to convert your assembly to. So if you're used to working with an older assembly of a species, and there might be a newer one, you might need to convert the IDs for that. And we also have the ID history converter. So with that in mind, I'm going to move on to a demonstration. So what we're going to do in this demonstration is we're going to go to Ensemble Plans. We're going to explore the homepage and find out what information we can find. And then we're going to look at a specific species. So in this case, we're going to look at wheat and find out what information we can find about the species, about the data that is available, and about the genome assembly itself. So remember, Ensemble doesn't do its own sequencing. The genome assembly is provided by other researchers around the world. Before I move on to a demonstration, are there any questions? Okay, so there's a question, and the question is, which tool does Ensemble use to generate the config? So the assembly is actually assembled by the people who submit the genome. So we don't assemble the genome ourselves, but you can find out how the genome assemblies were made by looking at the genome assembly page. So I will show you during the demonstration how you can find out that information. And then there's also another question, and the person is asking what Biomart is. Yes, this is a very good question. So Biomart is a really popular tool. We are going to look at it on Thursday. And it's just a tool in Ensemble on the browser that allows you to download Ensemble data in bulk. So loads of data at once. With any genomic resource, often it would be difficult to retrieve data because they're very big data sets. But with Biomart, you actually have a little user interface that allows you to just click and point with your mouse to find or to download that information. And then we have another question, and the question is, what's the meaning of comparative genomics? So comparative genomics is just one field in genomics. And what researchers in this field do is they compare different genomes against one another. So for example, by comparing the human genome against the chimp genome, and finding out how they differ, why they differ. They are also very useful for finding out the function of different genes. So if you're not sure what a gene does in a specific species, you could infer from a closely related species what it does in that species and so on. So that's one of the things you can do with comparative genomics. And then the last question I will answer for this just for time wise. Does Ensemble provide an option to get gene annotation file or GFF for specific species? We usually use UCSC Genome Browser. Yes, so you can download the whole annotation file in GFF or GTF even from Ensemble, and you do this via the FTP site. And again, I'll show you this during the demonstration. All right. Brilliant. So I can see there are loads of more questions coming in. I will get back to you guys on these, but just for now, I just want to show you a demonstration. Maybe some of my demonstration will answer your questions in this case. Okay, so let's switch to our browser. And I'm going to open a new tab and go to plant.ensemble.org. And that's ensemble without the E at the end. And I will also post the link into the Zoom chat. All right. So this is the Ensemble Plants website. This is the homepage. I will zoom in a little bit just to make it a bit easier for you to see on your screens. If it's still too small, let me know and I can zoom in a bit more. So this is the homepage for Ensemble Plants. At the top of the page or of the website, we've got this navigation bar. So this remains static no matter which page you're on. And these are just shortcuts to different popular tools and pages in Ensemble. So for example, we have shortcut to Hammer, which is a tool that allows you to find, to search protein sequences, so similar protein sequences. BLAST is another search tool which allows you to find similar nucleotide sequences and protein sequences. Biomart is another tool which we will explore more on Thursday. And we have a number of other tools also available which you can find under tools here. We also have this link that says downloads, and this is where you can find any whole genome data. So if you want to download the entire sequence of the wheat genome, for example, you need to go to our downloads for that. So I'm going to show you how this looks like in this page. So I've just clicked on downloads and I'm in the FTP download page. And if you scroll down, we've got this table that you can filter using this text box in the top right-hand corner. And this table gives you information about the different species we've got available. So that's what you're seeing in the second column, and the different files that you can download. So for example, if you want to download the entire sequence of Arabidopsis, you can do that by downloading either the DNA, cDNA, CDS, or ncRNA sequence. Oh, and also the protein sequence as well, actually, here. You can filter this table. So if you're looking for, let's say, wheat, so Triticum aestivum, you can just type it into the text box here in the corner, and that will filter your table down to any Triticum species we've got available. Okay, so let's close this tab. This is where you get whole genome data. So I'm back on the website now, on the homepage. If we continue on the navigation bar, we've got a few more options here. So if you click on more, you can find a link to our help and documentation pages. And there's also a link to our blog. So this is where we would announce any updates to Ensemble. We've also got a login page or registration page. So if you want to register an account with us, this is completely free. You can do so via this link. And this is especially useful if you use Ensemble tools. So if you use any Ensemble tools, we keep your jobs online for up to 10 days, and then we delete the results. But if you save your jobs into your account, your results will remain forever or until there is internet anyway. So if you want to keep your results from, let's say, Biomart or the Ensemble web, you want to register and save it into your account that way. Next, if we scroll down here on the right-hand side, we've got some highlights of any updates that we have. If you scroll down, we've got a section for the archive sites. So we keep archives open for up to 5 years, and then we take the website down. But the data is still available, so you can still get it from our FTP site. It's simply the website that's just not there anymore. And then we've got just some information about our funding partners. And if you scroll down to the very bottom, underneath here, we have links to our sister sites. So if you're ever interested in vertebrate genomes or fungi or bacteria, there are separate websites for that. But what I mainly want to show you here is on the left-hand corner here at the bottom, you can see which release you're currently in. So the release we're in is 59, and this was published back in May 24. So if you're using release 59 for your research, you want to try and keep it consistent, you can always refer back to down here. All right, so if we scroll back up, we've got, focusing on the left-hand side, we've got a link to the Ensemble Rapid Release website. So this is updated every 2 weeks as opposed to every 3 months. Again, we can see what release we're in, release 59, and some highlights to this release. So what has changed, what has been updated. And if we scroll back up to the top of the page, we have this section of the genomes we have available. So here on the right, we've got a list of favorite genomes. So these are just genomes that are most frequently accessed in Ensemble. But if you have an account, you can actually change this and change the order or even change the species on this list so that it's easier for you to access your species of interest. On the left-hand side, we've got a link that says view full list of all species. And I'm going to click this link now, because here you can find what species are available in Ensemble. So this takes you to the species page. Again, this is a table. And whenever you see a table like this, you can always filter it using the text box in the top right-hand corner. And you can usually also download the entire table. So there's this little Excel button here on the right. And if you hover over it with your mouse, you can download what you see. So if you apply a filter, you just download the filtered table, or you can download the full table if you like. So let's say you want to search for a species in here. You want to find out if your species is available. So you'll need to enter your species Latin name. So let's say Triticum aestivum. Triticum. So as you type it in, your filter or your table will filter down to include results for only triticum. And for each species, you can see we have some information available. So if we focus on the first line of the table, this is Triticum aestivum, this is wheat or bread wheat. We've got the classification, the taxon ID, the assembly name, so IWGSC, the accession ID, so wherever the sequence was deposited to in an archive. This is the original sequence ID that was given. And then we've got some other columns here, which tell you what type of data is available for the species. So for Triticum aestivum, we've got genetic variation data. We have some regulation data. So these include any microarrays, for example. We've got whole genome alignments, so comparative genomics data, any other alignments in peptide compara. So compara is comparative genomics. And this is just asking whether, this is just telling you whether any homology information is available and any pan-taxonomic compara. Again, comparative genomics, but across different taxa. And I'll talk more about this, I believe that's tomorrow, yeah. All right, so let's see what information we can find about Triticum aestivum. I'm going to click on the scientific name. And I'm just waiting for Ensemble to load. All right, if you ever see an error like this, all you need to do is refresh. It's usually a temporary server issue that's causing it. So I've refreshed my website, and this is now where I'm in is the Triticum aestivum species information page. So here you can find any information about the assembly. So remember, Ensemble doesn't do its own sequencing. We only take publicly available sequencing data and we apply our own gene annotation to the data. Oh, sorry, in this case, actually, the Triticum aestivum annotation is provided by the IWGSC, who are the original submitters of the sequence. So we've got information about Triticum aestivum, some general information, a description of the genome, a description of the species itself as well. Scrolling down under genome assembly, here's where you can find genome assembly specific information. So who submitted the sequence, who did the annotation, and so on. Under gene annotation, this is where you can find example genes and transcripts, which we will explore a bit later today. We can also see that we have comparative genomics data available for wheat, genetic variation, and some microarray annotation, so regulation data. And for each type of each data type, we've got some examples as well. So if you just want to explore how this looks like on the website, you can just click on the example regions or genes if you wish to do so. Okay, so let's look at information about the gene assembly, so the sequencing and the gene annotation. So if you go to genome assembly and click on more information and statistics, this will send you to the genome assembly information page. So this is where you can find a description of who provided the gene annotation and gene sequencing, and some more information about the gene annotation itself. So what algorithms were used and so on. On the right-hand side, we've got some summary statistics. So this is a bit more detailed information about where the sequence was deposited to, so where it was sent to, how big the genome is, who the gene annotation was provided by. So gene build is gene annotation. and this was provided by the IWGSC, or International Wheat Genome Sequencing Consortium. The gene build method, so the species in the Ensemble main site are annotated by us, whereas the species in the Ensemble Genomes sister site, so this is part of the Ensemble Genomes sister site, is the annotation is provided by different communities who work with the genome. And if you want to find the data source of the original gene annotation, there is a link here as well. We've also got some gene counts, so some summary statistics about the number of genes, number of coding genes, non-coding genes, and so on. Okay, so this is all I've wanted to show you for now. So I think now is a good time to move on to do some exercises. So if you go back to the training page, so again, I have this open still from earlier, but if you go to the training page, you want to scroll down to demos and exercises and open species and genome assembly. And we're going to use the exercises here. And the exercises are related to wheat in this case. So a few questions that lets you explore Ensemble and find out what information you can retrieve for wheat. In the meantime, I will be available in the chat for questions. So there are a few questions still that I haven't answered. So I'm just going to go through them. But if you have any other questions, please don't be shy and ask them away. You can enter them into the chat. You can also put them in a living document. And if you have any questions about the exercise, let me know. I can show you or walk you through how to do them if you have specific questions. I'll give you, I think, 20 minutes for this first exercise, just so that you have time to also explore the homepage and click around, see what pages we have, become familiar with the homepage. And then we're going to come back together and move on to the next module. So let me just check the time. It's 7:42 now in India Standard Time. So we're going to come back at 8 p.m. To continue on with the workshop. Thank you. All right, welcome back, everyone. I can see that some people have finished. That's amazing. Um, Before I ask if there are any questions, I just wanted to show you something. So I forgot to show you during the demonstration how you can find out if we have any other cultivars available. So let me show you on my screen now. I'm going to switch back to the Ensemble browser, or to my internet browser, actually. And I'm going back to the wheat species information page. So if you want to go there, you just need to click on the species name here in the top left-hand corner. And this sends you back to the species information page. And if we have cultivars available, you can find them here under genome assembly. So if you look at other cultivars at the bottom, we can see that there are 17 additional cultivars available for Triticum aestivum, and you can find a full list of these cultivars here. So let me open that page and show you how this looks like. So just a description of the different cultivars, who provided it, where, who sequenced it, and who provided the gene annotation. And if you scroll down, we've got these different cultivars available. If you're working with the newer RefSeq version 2 wheat assembly, this is also available via this page. So this is the RefSeq 2.1 version, whereas this one is the RefSeq 1 version. Okay. Apart from that, are there any other questions? I think I've answered all of the outstanding questions in the chat, and I have also answered all of the ones in the living document. But do you have any other questions before I move on to the next module? All right, no questions so far. Wonderful. So let's move on in that case. So for this next module, I'm going to talk to you about how you can visualize genomic regions and how you can find genes and transcripts in Ensemble. So I briefly mentioned that unlike the Ensemble main site, which is dedicated to vertebrate genomes, we don't actually carry out the primary annotation of the genes in the Ensemble genome's sister site, but rather we import the gene annotation from communities who provide the gene annotation for us. And these communities can use different ways of annotating the genome. So they may have automatic pipelines who do the annotation for them. So these would be different algorithms that annotate genes on the genome. They may also do manual curation methods to correct for errors in the automatic annotation, for example. Gene annotation can also be done by homology. So if it's a lesser studied organism, you can look at homologs of a similar genome, a similar species, and infer the function that way. Or they may also use ab initio predictions. So by looking at open reading frames in the sequence. And the annotation for ensemble genomes differs by the species. So depending on the species, a different institute does the annotation for us. So for example, for Triticum, we already know for wheat, the annotation is done by the International Wheat Genome Sequencing Consortium, sorry, IWGSC. And what they do is they use a combination of homology predictions and automatic annotation to annotate the genes. Or for example, for barley, this is provided by the International Barley Sequencing Consortium, and they infer the function of genes or annotation of genes using homology predictions and so on. So depending on the species, the gene annotation is provided by different institutes, and these different institutes can have different annotation pipelines to annotate the genes. Oh, sorry. So we either import annotations from specific communities or consortia who perform these annotations in collaboration with us. But we also import, or we mainly import genome assemblies from the International Nucleotide Sequencing Consortium, Sequencing Database Collaboration, INSDC. And this is just a collaboration between the European Nucleotide Archive, so EVI, the NCBI RefSeq, and the database of Japan, DDBJ. And these are how do you call it? Public archives where researchers can upload their sequences to. So because Ensemble is open source, the data that we display on our browser has to be open access as well. So if the data isn't deposited to the INSDC, we are not able to put it in Ensemble. So we have to make sure that the sequence or the genome assembly is just deposited to INSDC in this case. Okay, so in this slide, what you're seeing is what we call the region in detail view. And we're going to explore this during the demonstration, hopefully a bit today, but if we don't have time, we'll continue on tomorrow. And what the view shows you is just a visualization of a genomic region. So if you want to explore a genomic region on the browser, and you want to see what types of annotations you can find in the genomic region, you want to go to the region in detail view. So the region in detail view is, this might be a bit difficult for you to see, but I will show you during the demonstration. The region in detail view is made up of what we call tracks. And tracks is a genome browser specific term. And basically what it means is it's features mapped to a genomic region. So each of these lines represent different tracks. So the first track here are gene models. Then we've got the contigs track. We've got genes track. And again, another alternative gene model track here. And each of these tracks are a specific type of annotation in the genomic region. For each track, we also have a legend. So you can always find a legend at the bottom of the region in detail view. And you'll see that the different colors of the features that are mapped in the genomic region have different meanings. And we're going to explore this in the demonstration. So here it is again, a bit more zoomed in. Hopefully you can see it a bit better. So in Ensemble, a gene is defined as a collection of transcripts. You might also call these splice isoforms or isoforms. We use the word transcript, so that's what it means in Ensemble when you see that word. And there are different types of transcripts. And these transcripts are represented in these different colors, which you can find down here in the legend. Please also note that the colors can represent the different sources from which the annotation was imported from. In this case, for example, dark red and red are protein coding annotations from the IWGSC. And then this kind of blue color here are any genes from TGAC version 1. So that's another database providing gene annotation. The region in detail view is completely interactive. So you can click on the different features in this view. So for example, you can click on the track name to give you this pop-up window to find more information about the track. So what information you can find in the track or where it was imported from. There's also different options that you can do here. So you can delete a track, get rid of it if it's not information that you're interested in. You can also change the order of the track. So there is this kind of box next to the track names, and if you hover over it with your mouse, you can drag and drop the track to a different location. And you can also click on the individual features of the track. So if you look in the region in detail view, so let me just go back to the previous view. If you look at the region in detail view, if you see a feature like this is one genomic feature that is annotated. So this specific feature spans over this genomic region, and this specific feature is protein coding, and this is the ID for that feature. And if you want the exact coordinates, for example, or you want to find out more about the feature, you can always click on this feature, and you can find more information in that pop-up window. You can also change your mouse action on here. So in the view, in the top right-hand corner, there is two options. You can scroll with your mouse, or you can highlight a region with your mouse. And I'll show you how this looks in the demonstration. Okay, so let's look at the individual features of a transcript. So in the region and detail view, you might have noticed that a feature is kind of actually made up of these boxes and lines. And these regions just represent intronic regions and exonic regions in the transcript. So anything that's a box like this, those are exonic regions in the transcript. And anything that's like a line, like these, those are intronic regions. Now we also have empty boxes and filled boxes, like I'm highlighting here. And any empty boxes are non-coding exons, and any shaded boxes are coding exons. So we've got the 3' and 5' UTR. So this is the untranslated region of the transcript, non-coding exon. You'll also notice that the intronic regions, the lines, have a shape to it. So if you see a shape like this, so like a hill or like an A, that means that the transcript is located on the forward strand. And if it's shaped like a V, it's located on the reverse strand. You can also find out in which strand your transcript is located in by looking at the ID. So next to the ID, we always have this greater than or less than sign, and that just tells you the direction as well. So greater than is forward, and less than is reverse. Oh, and here I'm highlighting this again as well. So the filled boxes are coding exons. The empty boxes are non-coding exons or untranslated regions, and the lines are intronic regions. Okay, so I also want to talk about gene ontology or Go terms. So just like in any language, there can be multiple terms for a single biological function of a gene. So for example, one person might call it innate immunity, while another person would call it non-specific immunity. We, as researchers, know what we're talking about, but machines aren't that smart just yet. So to tackle this, the scientific community came up with Go terms, which is just a standardized way of describing a gene function to make sure that we're indeed talking about the same function, but also to make it machine readable. Go terms are made up of a unique Go ID, which make them more easily searchable, and also a human readable description. And you'll notice that Go terms are structured in a hierarchy. So a Go term can have a parent term, which describes the wider function of the feature, but it can also have child terms, so more specific, more detailed functions. And there are 3 categories, or we call them aspects of Go terms. So we've got the Go biological process. This describes what the gene product does. The Go molecular function describes how the gene does it. And the Go cellular component describes where is the gene product located in the cell. So for example, if we use the heat shock protein 101 gene product, and we were to look at the Go term aspects, so the gene product, what does it do? It's a cellular response to heat. How does it do it? By ATP hydrolysis activity. And where in the cell is it located? In the cytoplasm. So whenever we have these aspects available, we'll display them in Ensemble. Okay, so let's move on to another demonstration. We're gonna stay with wheat and we're gonna look at this gene in wheat. We're gonna visualize the genomic region and find out what information we can find about the gene and its individual transcripts. Now, let me just check the chat if there are any questions. So let me just move my window a little bit. So one question is, I'm working with animals, a mollusk species, but the species genome data is not available in Ensemble Metazoa. What's the alternative solution? That's a really good question. So if you can't find a species in Ensemble, have you tried Ensemble Rapid Release? If not, please try Ensemble Rapid Release. But if it's not available in Rapid Release either, you can always email us. So you can always email us for any species requests if you found that the species isn't available on our website yet. And let me send you the email address. So the email address you want to send is helpdesk@ensemble.org, and just tell us what species you want to include, and if there is a specific assembly that you're even interested in, and we can look into adding that to Ensemble. And then another question. So these things say only CDS or non-CDS, but not the actual function or name of gene. Please, can you comment? Yes. I'll show you now in the demonstration how you can actually find the gene function or description of the gene. Okay, so let's switch to our browser. And I'm also going to copy the gene name if you want to follow along on your screen. So let me just paste it into the chat. We're interested in Triticum aestivum, and the second line is the gene ID that we're interested in. So there are several ways to search for information in Ensemble. So if you're already on the species page, you can just use the species specific search. So just at the top of the page, you can enter your gene ID, click go, and that will send you to the specific page. If you're on the home page, so if you click Ensemble Plants, that brings you back to the home page. If you're on the home page, there are two different ways to search. Again, you can use the species-specific search, so that's in this green box here. The first thing you need to do is select your species of interest. So this is where the favorite species comes in handy if you want to change that to your favorite species. And then you can enter your gene ID in here, or you can use the side-wide search. So this here on the top right-hand corner. And this will search all of Ensemble plants for your gene ID. I always use the species-specific search, mainly because it's more efficient. It makes it a bit faster to search for any information this way. And you can also search for coordinates, for example. So all you need to do if you are searching for coordinates, all you need to do is enter the chromosome number. So this is just an example, chromosome number, followed by a colon, followed by the start coordinate, hyphen, end coordinate. So this is the structure you want to use if you're looking for coordinations, coordinates, sorry. But we have a gene of interest. So I'm going to paste our gene ID back in. And I'm going to hit go. And that will give me all of the results for the gene ID I just searched. So in this case, we only have one result. There's only one gene ID with that name. And this is it. So this is related to the species Triticum aestivum. And then the location is this. So these are the coordinates. If you want to look at the gene information, you can click on the gene ID. But if you want to go and just visualize the location, the genomic region, you can click on the location link. Let me click on the gene ID to show you what this looks like. So you'll notice in the top left-hand corner, you have your species of interest, Triticum aestivum, and then you have underneath these different tabs, what we call. And we've got one that's the location tab. We've got another one that's the gene tab. And then we've got another one that's the transcript tab. So depending on what data you want to find, what data you're interested in, you'll need to switch to the different tabs. For today's for today's demonstration, I'm just going to focus on the location tab because I won't have time to go through the genes and transcript tabs. But first thing tomorrow, when we come back to the workshop, I'll show you what information you can find here. Just to answer a quick question here. So one person is asking how you can find the actual function of the gene. So sometimes you would have a description in this part of the page. In this case, this is not available. So what you want to do is you want to make sure you're in the gene tab. And if you look on the left-hand panel, there is options for ontology. So these are gene functions. And here you can find the more specific functions of the gene if it's not available in the description. I hope that makes sense. If not, please let me know and I'll show you again. Okay, so let's focus on the location tab for now. So let me open the location tab. And you can see this page or this view changes completely. So depending on the tab, you'll have different types of views. If you click on the location tab, by default, you'll always land on the region in detail view. So that's what you're seeing here on the right-hand side. And this region in detail view is divided into 3 different resolutions. So we've got our coordinates, or you can see them up here as well. We've got our coordinates, and it's showing you the coordinates in relation to the chromosome. So this is a chromosome overview. And our coordinates that we search for is highlighted in this kind of red line here. Then we've got the region overview. So our region of interest, again, is highlighted in this kind of red box here. So it's a very thin box, mainly because it's a very short feature, very small feature. And if you scroll down, we also have what we call the region in detail view. So just as the name says, it's showing you your region of interest, but in more detail. Let's just focus on the region overview. So for the region overview, you can find at the top of the view, we have got a scale. So this is showing you the region in 50 kb upstream and downstream. And our region of interest or our gene of interest is actually highlighted in this green highlight here. So that's our gene of interest. And our gene of interest is this little box here that you can barely see. So this is why the region overview is more, region in detail view is more useful here. But as mentioned in my presentation, this view is completely interactive. So you can use your mouse to drag and drop and highlight any regions, or you can scroll forward and backwards in the genome to look at the neighboring regions. If you want to do that, in my case, you can't really see it here, but you should have options in the top right-hand corner to change your mouse action. So for me to see this, I'm going to zoom out a little bit. I'm sorry, you won't be able to read much on my screen, but I mainly want to show you how this looks if you wanted to change your mouse action. So once you zoom out enough, you can see these options. So the first one is to scroll. So if you click that, your mouse you can use your mouse to scroll back and forth in the region. And underneath, you can see it will ask you to either, if you want to update the image, so update it to the region that you've highlighted here, or if you want to reset it, so have it as a default that the original coordinates we searched for. If you want to mark a region, you need to change your mouse action again, so to this rectangular box. And what this allows you to do is to click and drag your mouse. And when you let go of your mouse, you can either mark the region, so this will put a highlight in that region, or you can, what's the other option? Jump to the region. So if you jump to the region, this will update the region in detail view. So you can click on the individual features. Oh, sorry. If you want to click on the individual features, you need to select the scroll option for your mouse first, and then you click on the feature. And then you can find in the pop-up window, you can find more information about whatever feature you just clicked on. So the one I just clicked on is the gene we are actually looking at. And this is the gene ID. We've got the GC content, what type of gene it is, protein coding, which strand it's located in, and so on. So let me close this and zoom back in so you can see it a bit better. And I'm also going to reset my configuration. So if you reset your configuration, that will just get rid of all the highlights that you've made, and it will scroll back to the actual region that you're interested in. So in our case, our gene of interest. We reset configuration. So here we go. We have our gene of interest. That's this little tiny box here. And this is the label for that region. Okay, if you scroll down to the region in detail view, we can see we've got a few tracks already turned on by default. We've got the chromosome band, contigs, genes, alternative gene models, and variants, so genetic variants in this region. As mentioned, you can change the order of these. So if you wanted to, for example, see what variants are located in exonic regions, for example, so these boxes, you can hover next to the track name and click and drag your mouse and move it closer to your other track that you might be interested in. Again, this is completely interactive. So you have your mouse actions again available in the top right-hand corner. You can click your region or your feature of interest to find more information about it. And you can also delete any track by just clicking on the track name, clicking on the track name, and clicking on the X here. So these are only a few tracks that are being shown, but you can actually add more tracks to this view if you're interested in any other types of data. So if you go to add remove tracks in the top left-hand corner, or if you go to configure this page in the far left panel, both of these buttons do the same thing. So they open this pop-up window to show you all of the available tracks we have in Ensemble. So these are divided into different categories. That's what you're seeing here in the left-hand panel. And on the right, you can see the different tracks we've got currently turned on. So for example, the contigs track If you see a little symbol in this square, it means it's turned on. And if you want to find out what information you can actually retrieve in this track, so what genetic feature you're looking at, you can click on this eye icon in the far right, and that's just showing you a brief description of each of the tracks. If you're interested in specific tracks, for example, you can also search for a track name. So for example, we can search for maybe, trying to think of like an example. For example, you're interested in variants from the serials DB archive repository. You can just type in serials DB, for example, and that will filter down all of the tracks with the term serials DB. Well, going back to active tracks, if you want to turn a track off, just click on the square next to the name. You just click turn off, or you can also change the style that you want here. Okay. And then finally, one more thing before we leave it for today. You can download or you can export the image if you want. So you can use these images from Ensemble in your reports. You can click on export image up here. So this will export the region overview. If you click export image down here, this will export the image for the region in detail view. And if you want to export the data, so for example, the annotation or the sequence of your region of interest, you can click on export data in the far left panel. And that gives you, again, a pop-up window. You select what type of format you want. So if you want the sequence or if you want the gene annotation, you can also highlight if you want to visualize any up and downstream regions and just click next, and that will download it to your local machine. Okay. So with this in mind, we've looked at the genomic region. We know now how to visualize the genomic region. Tomorrow, we're going to continue on, and I'm going to show you how you can find out information about genes and transcripts. So what function you can find, links to other databases, for example. We're also going to explore how we can find IDs for certain enzymes. So there's a question in the chat. So hopefully tomorrow you'll have an answer for your question there. If not, please post your question again, and I'll answer it after my second part of the demonstration. So for now, I hope you have a nice evening. Thank you all very much for this amazing first session. Thank you for all the good questions that you asked. If you have any questions in the meantime, please feel free to put them into the living document and I will answer them as they come in. So I believe it's the same link for tomorrow, and I'll see you again at 7 p.m. Indian Standard Time. Thank you.
So thank you, ma'am. Thank you for your wonderful presentation on Ensemble Plant Genomic workshop. And we believe our participant enjoyed your session. We will meet again tomorrow at the same time, 7 p.m. Indian Standard Time. I will also share the video recording of this session as soon as possible. Thanks, ma'am. Now I seek your permission for ending today's session.